So my name is Stamatis Papthanasiou. We're now for a bit over a couple of years in Germany. We set up my lab in Mainz on the Institute of Molecular Biology. Before that, I was in Boston, the lab of David Pellman. And the point today is to just give you a hint of what we are up to now in Germany. But the main part will be the work, my postdoc work that I did with David in Boston, which was basically the basis of most of the things that we're doing now in Germany. And maybe you noticed already, I changed a little bit. I changed a little bit the title with introducing this term here, chromosome PTSD, just to make it a little more like catchy. Chromosome post-trauma stress disorder, because this is kind of how I view what we're doing in the lab. We are kind of going after whether there is any memory of a trauma when a chromosome is actually undergoes stress or trauma or an insult, whether there's any memory of that in the next generations, and you will see where I'm getting with that. Okay, so we're interested in mitosis, especially abnormal mitosis, and this is just to make the point that, of course, it's important, it's fundamental for the cell to undergo error-free mitosis, and that's why the cells have evolved mechanisms to ensure that cells that accumulate mitosis for several different reasons. Here I'm just showing a couple: when you have abnormal number of centrosomes, or when you have merotelic or syntelic attachments for whatever other reason. Cells have evolved mechanisms to ensure that those cells will stop dividing further, will arrest. But that's not always the case. Cells escape checkpoint mechanism when they do mistakes, and cells do mistakes when they divide. And the hallmark consequence of abnormal mitosis, of errors in mitosis, is the generation of aneuploidies, when the daughter cells end up having more or less copies of a chromosome or a chromosomal segment. So another hallmark consequence of abnormal mitosis, of mitotic errors, is the generation of abnormal nuclear structures. And here I'm illustrating two of the most recognizable structures, which are micronuclei at the top and chromosome bridges at the bottom. Micronuclei are formed when you have a chromosome lagging in a mitotic, and that mitotic exit can be encapsulated in its own nuclear envelope. And this is literally a small nucleus, a miniature nucleus next to the primary nucleus in the daughter cell. Chromosome bridges are when the ascending chromosomes are formed, and then upon mitotic exit, you have this thread of DNA connecting the two daughter cells together. And the point that I want to make here, although I will not be talking almost at all about chromosome bridges, is that the more we study these structures, chromosome bridges, the more we realize that they share significant similarities to the biology of micronuclei. So these are kind of sister abnormal nuclear structures. Okay, so micronuclei, chromosome bridges, together with other forms of nuclear deformations like nuclear blebs or buds that you see here in this DNA stain of a cancer patient. They're all fall under the umbrella of the so-called nuclear atypia, which is a hallmark in cancer. Pathologists have been used nuclear atypia for more than a century to assign tumor grade and cancer patient prognosis. So this kind of old knowledge, it's known for many years that these structures are prominent in cancer. What is new and what is really getting very exciting is that the last years, the last like 15 years or so, we have realized that these structures are not only a consequence, like a phenotype, a biomarker in cancer, but they are also the cause, the origin of genomic instability. So these are basically the mechanistic origin that explain ongoing genomic stability and can generate continuously evolving cancer genomes, like the one that you see here of a cancer patient. And we now know that these structures are also the mechanisms behind very prominent rearrangement events that we now see in cancer genomes from work from Isidro and others, we know that phenomena like what I'm showing here, chromothripsis, are very dependent on cancer, and these structures, macronuclear chromosome bridges, are the mechanistic origin of those events. I will not talk in detail about that. Of course, we heard a lot about chromothripsis from the previous speakers. I will mainly focus on the cellular events that lead to this phenomenon like chromothripsis, and this slide basically summarizes what we know about in general, what we know about the cell biology of these structures. So macronuclei are very abnormal nuclear structures in abnormal nuclear environment. Everything you study, it's pretty abnormal macronuclear. DNA replication is severely affected. Import is abnormal, and this we speculate is because we kind of know from work from David and that nuclear envelope assembly is severely abnormal, and that's the reason that import is defective in these structures. And from seminal work from Martin Hetzer and more recently from Samba Kum and Stefano Santaguida, we know that these structures undergo irreversible spontaneous rupture events during their lifetime. And because of this rupture, DNA damage is accumulated in these specific chromosomes or chromosome segments that are in macronuclei. And there's another wave of DNA damage that's acquired in the subsequent mitosis. And eventually, this chromosome or chromosome segment is fragmented. And that's the reason that in the next cell division, in the next interphase, we end up having this rearrangement signature after these fragments are repaired. So there's a lot of super cool and interesting biology here, and very high impact papers that came out the last years. I'm just summarizing some of them here. I will not get into details on that. I just want to make the point that most of the biology we know about these structures is focused on this generation, what I call generation 1, which is basically the interface after the initial event, the initial mitotic era, when the macronuclei are generated. There's not that much information. It's still kind of a mystery about the biology of these structures or these cells after the next generation, or better, after many generations. So this is exactly what was my question. So we know basically that this introduction was just to make a point of what you know, that micronuclear chromosome bridges lead to genetic instability. So that was exactly the point. When I joined David's lab, we wanted to go beyond the first interface, beyond the interface where macronuclei are introduced, and ask the question, what happens in the next generation? And this is what I'm showing in this illustration. Especially what happens when this abnormal chromosome reincorporates back to the primary nucleus of a daughter cell. So there are two possibilities, two options. Either the macronucleus chromosome will persist as a macronucleus in the daughter cell. I'm not showing that here. Or it will reincorporate back to the primary nucleus. This is what I'm showing here. This was kind of the focus of my study. What happens when this abnormal chromosome is back to the normal nuclear environment, where enzymatic machinery is available, because remember, import is defective in macronuclei, many enzymes are absent. So it's interesting to study what happens when this abnormal chromosome goes back where in the normal nuclear environment setting. And I asked the question, what happens beyond the genetic events? So we know that macronuclei lead to genetic events, but what happens, what are the functional consequences? And especially I focused on the gene expression, transcription of these chromosomes, and on chromatin state. So I will first start with the first part, the kind of easier part, what happens when the chromosome is in the macronucleus, before going to the interesting and cool question, what happens upon reincorporation. So the first part is about studying the transcription in chromosomes when are in macronuclei. And I wanted to take a genome-wide approach to study that more in detail. People have had already investigated that by immunofluorescence approach, and there was evidence that transcription is defective, but nobody had done a detailed investigation at especially genome-wide. So I wanted to to develop a single-cell RNA sequencing approach to study that. And of course, that was not very, very easy. There were multiple challenges on the way. And I think it's informative to go over a couple of those challenges and what we did to overcome these challenges. So the first challenge was, how do we know which chromosome is in the macronucleus? How do we infer the chromosome context? So to study the transcription of the specific chromosome by single-cell RNA sequencing. And the idea that we had back then was to study the family of the macronucleus cell. In the case of generation 1 pairs, to study the sister of the cell after the initial mitotic event. And the point was that we can infer the identity of the chromosome macronucleus because we know what to expect from the chromosome segregation scenarios. So these are the two segregation scenarios, the normal segregation scenarios at the bottom, where macronucleus will be formed in the correct cell, and then we'll end up having copy number 2 for this chromosome in both cells, or the abnormal segregation scenario where the macronucleus will be formed in the wrong cell, and then we'll end up having an imbalance, a trisomy for this chromosome in the macronucleus cell and a monosomy in the sister. So basically, if we manage to sequence this pair, both the sister and the macronucleus cell, and we find a monosomy in the sister, then we know that the macronucleus is this exact chromosome, but the other haplotype, in this case, the full magenta haplotype in the macronucleus cell. So this was the idea. These tricks like that we used to infer the identity of the chromosomes in macronuclei when we induced random chromosome segregation. And this is the main way that we generate macronuclei in the lab. But how do we sequence the family? How do we do that? Nobody was able to do that before. So again, I'm making the very long story short. We had to develop a new method. And in the work we published with David, we called it LuxEC2, which is a much better name of what I choose to call it now. We're trying to rebrand it with this very, very ugly Greek name, Simati. You can put in the chat if you like it, or don't put it if you don't like it. You'll hurt my feelings because kind of end up with that. So same cell imaging and direct isolation of single cells for subsequent genomics. So basically what we're doing here is we can link phenotypes with single cell genomics. We combine live cell imaging. We observe the cells under the microscope. So in that way, we know exactly the cellular events that happen with the use of specific fluorescent markers. In our case, in this work, we followed macronucleus generation, and we followed rupture events. And we can also track the cells, track generations and lineages, so we know the cell relationship information. We know exactly which cells are sisters, cousins, etc. And by retaining the history of events and the cell relationship information, we capture specific cells of interest directly from the live cell culture. This is very powerful, and then we can proceed with subsequent genomics. In this case, we did single-cell RNA sequencing. So this is very, very powerful method. This is now a patent since last summer. I'm very happy to get into details with whoever is interested. But I will just jump to the data, how they look like. So these are two examples. At the top, you have a case of abnormal segregation, at the bottom of a normal segregation. And what you see here is single-cell RNA sequencing data for each chromosome, and with the two, with the dark and top and white there are the two alleles. We have phasing. We were able to phase the transcriptome in this cell line. These are RP1 cells. And immediately what you realize, what you can get here is that you see the exon activation really nicely appearing. This cell line is female cell line, and also a known event, the only event in RP1 cells, a 10q duplication. And this is real single cell level. This is not aggregating thousands of cells. This is really real single cell level information. And the only other chromosome that you see imbalance here, transcription imbalance, is copy number imbalance, is chromosome 2. And in this case, the sister is monosomic for chromosome 2. And we know that this is the sister because we imaged these cells. It didn't have macronucleus, and this had macronucleus. And so we know then that we infer that the other haplotype of the same chromosome, of chromosome 2, the magenta haplotype, the full magenta haplotype, is the micronucleated haplotype. And we expect a 3-copy number here, and we've got 2, so that's how we inferred that this chromosome was a macronucleus and was transcriptionally defective at the whole chromosome level. So this is kind of, I just want to show you how we kind of approach this experiment and the methods that we used. And I will open a parenthesis here because this is a new experimental approach, a new method, a new technology, and a new bioinformatic pipeline that was basically magic from Cheng Zhongzhang in Boston. So we wanted to benchmark both the experimental approach and the bioinformatic new pipeline. And to do that, I wanted to really target a specific chromosome macronucleus. This was kind of the first thing that I wanted to do when I joined David's lab. That was kind of the holy grail back then in the field. And we ended up doing it with a very, very simple idea. That was 2015. What if we cut what if we introduce a very efficient cut before mitosis? Maybe we generate an acentric chromosome fragment that will encapsulate itself in a nuclear envelope and will form a macronucleus. And this is exactly what we managed to do. Here we are targeting chromosome 5q, and you see by FIS here 5q is in macronuclei. And now we have a method that we can target specific chromosome arms in macronuclei. And this is what Isidro mentioned in the beginning. We published this work as one of the first on-target side effects of CRISPR. So whoever is CRISPRing cells, you might be introducing on-target micronucleation. But for me, that was a method. I now have my target macronucleus approach, and I'm introducing that to the same cells, RP1s. And here you see a pair, and the only chromosome that stands out is chromosome 5q. And if you get into more high resolution, you see that exactly where I'm cutting, exactly where the macronucleus was formed, the haplotype that was in macronucleus is transcriptionally defective. So we used it as a means to benchmark experimental bioinformatic approach. Going back now to the random macronucleation and the main data set, this is the bottom line result. The majority of macronuclei are transcriptionally defective, except for a very, very small subset of intact macronuclei that didn't rupture, that expresses in normal levels at the whole chromosome arm resolution. So basically, this was confirming that transcription macronuclei is severely affected. And we also carried out chromatin characterization, and we found that repressive chromatin marks are enriched in a subset of macronuclei, but more strikingly, open chromatin histone modifications like K27 acetylation, H3 K27 acetylation are severely decreased in macronuclei. So this was kind of in agreement with the transcription data. And I have to say that at the same time, Sambakum published a beautiful paper, mostly focusing on the chromatin characterization in macronuclei. Okay, so again, I just want to introduce you in this part the methods, the approaches that we took, but let's go to the really, really cool question. What happens after this abnormal chromosome? And remember, most of these do not express in macronuclei. What happens after these macronuclei go back? These chromosomes go back, reincorporate to the normal nuclear environment. Everything is all the enzymatic machinery is available there. So this is how it looks like from the imaging of this of the SIMADI part, the imaging part of the SIMADI approach. You can you start with 2 cells, one with macronucleus, and this is the sister cell, and then at some point this will divide. First, we tracked the rupture events. I cannot drag this video, so sorry, you have to watch the whole thing, and I think it's pretty big. So at some point, macronucleus will rupture. Here, we can observe rupture events. You can introduce whatever fluorescent markers. You can have cellular event information, and then the sister divides. So we have what we call the niches. And then at some point, the macronucleus cell divides. micronucleus chromosome is reincorporated because you don't see micronucleus anymore at the daughter cells. So basically, we end up having the 4 cells after the initial mitotic event. So we now can capture the whole micronucleus happy family, and we can with the same tricks, with the same idea, trying to infer the identity of the chromosome that was the micronucleus, taking into consideration the chromosome segregation scenario and trying to figure out what happened. Now it gets a little bit more complicated, but it's again the same idea. At the left, you have the normal segregation scenario, and due to the allelic imbalance, the copy number allelic imbalance that you will have because of replication defect micronuclei, you expect to have disomic cousins and 1 to 2 copy number for the daughters. In the abnormal segregation scenario, you expect to have monosomic cousins and 3 to 2 for the daughters. Doesn't matter, these are details, but the idea is that we try to infer what happened, the history of events based on the data. And it worked beautifully. This is just one example, just to show you how it looked like. It's kind of a complicated example because there were 2 micronuclei, 2 chromosomes in this case. So just focus on chromosome 5. So the cousins here were monosomic, so that's an abnormal segregation scenario. Immediately we can infer that was an abnormal segregation scenario. And then we expected a 3 to 2 copy number for the daughters, and we see 2 to 2. So we kind of infer in this case a transcription defect in the reincorporated chromosomes. But this is the important plot. So what we found is that intriguingly, most of reincorporated chromosomes, micronuclear chromosomes, will recover transcription. Remember, most of them, almost the vast majority is transcriptionally defective when they're in micronuclei, but upon incorporation, most of them will recover, except for a subset of micronuclei that ruptured the previous interface at their lifetime, and then we observe inheritance of the transcription defect, which was, from my point of view, both results are really, really intriguing, are really, really interesting. Both the recovery of transcription in the majority of reincorporated micronuclei, but also the inheritance of the transcription defect on a subset of micronuclei. So again, novel method, single-cell based, single-cell RNA sequencing, novel technology and bioinformatic pipeline. We had to convince ourselves and everybody with orthogonal approaches, and this is something that was fundamental for our work, and we love setting up new systems. So here I'm going to the kind of the second holy grail for me during my time in Boston. I really wanted to be able to visualize the chromosome, the reincorporated micronuclear chromosome in the primary nucleus of a daughter cell. So I developed a couple of systems. I will just show you here. I will start with that, which is basically based on an old system that Robert Spector developed back in the days, 2004, and kindly gifted us. And we heavily re-engineered the whole thing. The idea here is that you can visualize the genomic locus of this transgene, which is very importantly integrated in a unique integration site in chromosome 1p. So you can visualize this specific locus because it harbors a series of LACO repeats. And you can visualize the nascent transcripts because the RNAs that are expressed have MS2 aptamer loops. So by the use of LACI fluorescence with fluorescent markers and MS2 code protein with fluorescent markers, we can visualize both the genomic locus and the transcription of this locus. And again, we re-engineer the whole thing, and now we have a method, we have a system, that you can see chromosome 1p in micronuclei, and you can see these chromosomes, chromosome 1p, that are in micronuclei and the transcription in this channel. And I will start the videos here. Both cells will divide. First, the top cell will divide. Here you can also observe rupture events. So first, the top cell will divide at some point. I think somewhere around here. Okay, first you have to see the rupture. Yes, we have. And then the top cell divides for the third time. Here, yes, top cell divides, reincorporates. You see the reincorporation because we contract the genomic locus, and there is no transcription in this chromosome. Bottom cell divides, reincorporates, and there is a massive burst of transcription exactly upon mitotic exit, and the normal level transcription during the next interphase in the cell. So basically, you see both flavors of transcription recovery upon incorporation or inheritance of transcription defect upon incorporation for the macronucleate chromosome. And here you see quantification of these cases and the aggregate plots here. So these are the recovery cases. In light gray at the back, you see these are controls. And you see the case of recovery in the cases of transcription defect upon incorporation. This is only tracking the 1p chromosomes that were macronuclei and now are incorporated in a daughter cell. And this is the bottom line plot. And I think you can appreciate that it's really looking like a mirror. It's really a mirror image of the sequencing data. We're very, very happy seeing that. The majority of reincorporated macronuclei recover transcription, except for a subset of rupture macronuclei that inherit the transcription defect. So next question was, okay, how is that happening? What facilitates this kind of, what's behind the inheritance of the transcription defect? That was really, really intriguing for us. And the first thing that we immediately thought was that the chromatin alterations that we found that are present in macronuclei are inherited in the next generation. And this is what we followed up. I started actually with a very, very simple experiment. At the beginning, I followed cells by live cell imaging after macronucleus generation and on a gridded cover slip. And then after the macronucleus were incorporated, I knew exactly where the daughters are. And I fixed this disease, and I performed immunofluorescence. And I went back to see exactly the daughter cells. And I did immunofluorescence for gamma S2AX. And the striking result was that the majority of these pairs that I knew were daughters upon reincorporation, macronucleus reincorporation, one of the two daughters had this bleb of gamma H2AX that is really, really striking, like a chromosome territory. And immediately I was like, okay, this has to be the macronucleus that is reincorporated. But of course, I had to show that. And that brings me to the second system that I call MNTRAC, that we developed in order to visualize the reincorporated macronuclei. So this is based on MDC1, a fluorescent version of MDC1 in RP1 cells. MDC1 is recruited in gamma S2AX sites. And here you see again, we can follow rupture events, and the macronucleate cell will divide here and reincorporate. Exactly upon mitotic onset, you see accumulation of MDC1 in macronucleus, and then we can track the reincorporated macronucleus in the primary nucleus of the daughter cell. You see this bleb of MDC1 here. That was really, really exciting to see. And if you don't believe me that this is macronucleus, maybe here with this video, I will convince you. We kind of prove that in many different ways, but I think this is pretty informative. Exactly upon mitotic onset, you see the correlation with MDC1, specifically of the macronucleate chromosome. And then you can track the reincorporated macronucleus in the primary nucleus of a daughter cell. So now we both managed to have a system that we can track the reincorporated macronucleus, but also we proved that the damaged chromatin state the damaged chromatin state of macronucleus can be inherited in the daughter cells, can be inherited in the next generation, which was very, very important discovery. So here you see how this reincorporated macronuclei, damaged macronuclei look like, and we call these structures MN bodies. In this case, these are the damaged MN bodies because we use MDC1 to detect them. And now we have our system. We, of course, validated our main findings with this system. We confirmed that by IEF approaches that transcription is defective by staining for phosphoserine 5, for RNA pol2, and H3K27 acetylation is also decreased, which is very, very intriguing result, meaning that the histomodification alteration that we found in macronuclei is inherited in the next generation in the case of MN bodies. Okay, so I will stop here on this work, and basically the model was that in macronuclei the chromosomes are severely defective transcriptionally, and also the chromatin state is altered, specifically H3K27 acetylation is decreased. And there are two possibilities: either there is no damage and then the defects will recover. So transcription and chromatin state will recover in the next generation after incorporation. But if there is damage accumulation on this micronuclei, then this damaged chromatin state can be inherited, and because of that, transcription and chromatin state will also be inherited in damaged. And we showed this correlation of damage and inheritance of the defective chromatin state and inheritance of transcription defect. I didn't show here the data, but we saw this correlation with a couple of approaches in this work. And I want to make the point here that most of our work focused on this specific interface after the reincorporation of macronucleus. So the generation 2 of the after mitotic event. What we are really focusing in the lab now is to go beyond that and ask the question: what are the long-term consequences? What is the long-term memory of these insults? And just to give you a hint, I will not show much data. Okay, this is super fast, and I cannot slow it down. But here you will see a macronucleus reincorporated. Here you see MN body. And a lot of things going on here that we try to understand. But here I just want to make the point that this cell will divide again. And MN body will be formed here, divide again, and MN body will be formed in the next generation as well. So this is generation 3 MN body. And this is just to make the point that these structures can be long-term transgenerational inherited in more than 2 cell divisions. Okay, so this is exactly what we are focusing, and this is where I'm getting with the chromosome PTSD. There's a trauma, there's an insult in a chromosome, there's an abnormal chromosome. Is there any memory of this insult in the next generations? And in the lab, we mainly focus on the potential contribution of non-genetic mechanisms, of non-genetic effects that may act together or on top of the genetic effects like chromothripsis rearrangements, and might lead to selective adaptivity and tumorigenesis. So this is completely a mystery. It's a complete black box. What is the contribution of non-genetic functional consequences that are introduced by mitotic errors? And we are investigating that in several different layers. So we are asking questions on transcription dysregulation, epigenetic state, 3D chromatin dysregulation, how is chromatin architecture altered, affected in these abnormal chromosomes, and how having an MN body, how does that affect the overall nuclear chromatin architecture 3D organization, whether this all lead to epigenetic instability, nuclear structure abnormalities, and eventually the selective adaptivity and tumorigenicity. These are the questions that we're going after. Technology development is very important in our lab, but we have ways now that we can track these structures. We can permanently tag chromatin of these abnormal chromosomes independently of the DNA damage state. And this I didn't show you, but this is a very cool method. I'm happy to talk more about it. We have the system that we continuously continue to evolve here to target specific chromosome, to design specific chromosome for misregulation and for MN body formation. And of course, we have our SIMADI approach that we can select cells of interest for subsequent genomics. And just for the last couple of slides, I want to just tell you a little bit more of what we're doing here on this aspect on the SIMADI part. We kind of now are in the next generation of SIMADI. We have developed the state-of-the-art single-cell RNA sequencing assay, SMATSEQ3, which is working really well in the SIMADI pipeline. And with that, we hope to answer the question of transcription dysregulation in MN bodies. And from the heroic efforts of Ruxandra in the lab, we now have evidence that we can do the SIMADI high-C, single-cell high-C, with this SIMADI approach. This is all integrated in our SIMADI pipeline. And I'm pretty sure you are all thinking that this is really long shot kind of assays. These are really difficult single cell based assays. How can we ever get the number of cells to try to get an idea of try to get an answer of what in our difficult questions that we're asking, and I totally agree. So we kind of heavily invested the first couple of years in kind of upscaling this method, and now we have a pipeline that it's basically semi-supervised linear tracking, transrelational linear tracking that is fully integrated in SIMADI. What we are doing is that every frame that we image in the microscope downstairs, it's communicated directly in a computer in the lab. So it's on-the-fly imaging a real-time analysis. And then we performed machine learning-based tracking with crazy important help from our collaborator, Martin Weigert here in Germany, who is a cell tracking magician. So we're using his algorithm, and we are segmenting and tracking the cells in real time. And then we have a user interface that we can correct lineages, and we can select cells for specific cellular events. And then we end up, before the imaging is done, we end up with specific coordinates of the lineages of the families or the cellular events that we want to capture in our SIMADI pipeline. And this is not very easy. It's like I cannot show you here, but anyway, we're using these RP1 cells that are really the worst cell line to use for this kind of tracking, because you see how crazy, how maniacs these cells are. Here, we're really getting all these algorithms to their like potential. Okay, so that is my last slide. This is just to give you a hint of what we're up to. We think that with this direction, trying to understand non-genetic effects from that originate from mitotic errors, we might contribute in the understanding of this elusive phenomenon of epigenetic heterogeneity that's observed in cancer. And eventually, hopefully, the dream would be that maybe we even unveil approaches for new therapeutic opportunities. And with that, I would like to end with thanking the lab. Now we are actually a real lab. Up to like a year ago, we were only like 2 people. So I'm very, very excited. We have the amazing, the best people in the lab, but we are recruiting, and in every level, please email me if anybody is interested. Funding, and of course, most of the work that I showed you, the previous collaborators, David, of course. And thank you all for the attention.
Thank you, Stamatis, for a terrific talk. Any questions from the audience? Please raise your hand or write them in the chat. Yeah, perhaps I will start while others basically think about their questions. So, what is the throughput at this stage, Stamatis, of the SIMADI platform, and what are the practical bottlenecks of scaling up?
Okay, since you remember the name, that means that it's not that bad. Okay, so it's literally the last couple of months that we managed to have successful experiments with this pipeline, the AI SIMADI that I presented, and with this, it's totally different level. We can go up to, at this point, 8 cell states easily. We can track cells 8 cell states, and we can capturing is not the limiting factor here. We can capture easily dozens of cells per ring that we are using to capture the cells, but you can have multiple rings for this experiment. So capturing is not the limiting factor. The limiting factor is really the tracking. If you are studying a rare event like us, this is the limiting factor. Mm-hmm. So finding the cells of interest, the limiting factor. The capturing in our pipeline is really, really easy. You can go hundreds.
Yeah, I mean, that's indeed very, very powerful. I guess another question. So when you basically follow the cells over 2 generations, and then you saw the changes in transcription, were there any recurrent changes or pathways that were affected, or basically, did you see any changes in proliferation or fitness in the resulting cells after 2 generations? I mean, was there any? Yeah, exactly. In the 2nd generation, was there any? I mean, that's perhaps too early to say, right? But any changes in proliferation, cellular fitness, or? recurrent transcriptional programs? Yeah.
Great question. This is something that we didn't have the data from this data set that you saw before, because we didn't end up following after the next division. I was capturing the cells during generation 2. But this is exactly what we're trying to do now. We're trying to go to the cell fate of these semen body cells and what happens to fitness and selective adaptivity when you have specific chromosomes there, etc. So this is exactly what we're trying to do now, unfortunately. And I will have to start answering, yeah, I don't know, we're doing that, but that's actually true. Give us a little bit more time.
Fantastic. We'll indeed keep an eye on that. So we have a question from Nalini Kolar in the chat. So a more specific question: when micronuclei are reincorporated back into the primary nucleus, do they affect PML bodies?
We haven't studied that, and I don't have any indication towards that. But this is something that we haven't studied.
Yeah, in all positive cells. Yeah, that's okay. Yeah, thanks a lot for the question. Florian, do you have any question? I see you around.
No, I just wanted to make it a bit more personal here. We got 50 people. Don't be shy. We're all nice. How long can we wait?
I'll ask a question.
Hi, Pipa. Go ahead, please.
Hi. I'm really intrigued by the DNA damage data, and I was just wondering if you were able to follow that and whether the DNA damage is repaired more slowly compared to the regular nucleus once it is reincorporated, or did you just look at a single time point?
Well, that's a great question. We didn't really focus on that during this work, and in the lab now, it's not the priority to do that. I know other labs are working on that. There is indication there are some issues in the. You asked about replication or repair? Sorry.
Repair.
Repair. No, we are not really following that in the lab right now. I don't have a good answer for you, but this is definitely something that is important to follow up, especially since we have now the tools to follow these abnormal chromosomes.
Thank you.
But Peter had these beautiful papers that he kind of addressed. And it's important. We know that now timing of repair and exactly what happens in specific context of human bodies. These are really, really interesting questions, really cool questions.
I'll say one question from my end. So can you please elaborate a bit more on the on techniques that you use to track basically the micronuclei? You were saying they were in some dyes. Can you please give us a bit of a summary of that?
So the MTRAC, the main method that that we used, and the main method that I showed you today is this MTRAC. With this is a simple cell line. This is MDC1 SNAP. We just have a fluorescent version of MDC1. The thing there is that MDC1s accumulated in micronuclei upon the next cell division, when you have basically the envelope collapsing, and then you have the human body. We don't see MDC1 micronuclei, and I think this is the reason that previously the cell line existed before, a similar cell line, but previously this was not being used for tracking micronuclei, for tracking human bodies. So this is a very simple cell line. We also have a methods paper now out. We are describing exactly how we made it, but I'm very happy to share the line as well.
Any more questions for Stamatis?
I didn't get any comments about the name, but apparently it was all negative.
No, it's very nice. Yeah, no, we're still focused on the name. I really like it, to be honest.
It's a thing Greek people have. They have to give Greek names to everything,
Non-Greek people as well.
True.
Look at the terms in this field.
Exactly.
Yeah.
It's full of people. I mean, at least this is your language. At least you understand what you're saying. All the others just make up these Greek names.
It's actually a lot of Greek people behind the Greek names as well, like Chromothripsis as well. There is a Greek person behind the name Chromothripsis. There is. Ask Peter Campbell.
Okay.
Yeah. Then if one starts, then everybody has to follow with chromo something Greek, if one starts.
That's right.
Okay, thank you so much. It's been a great pleasure.
Yeah, thanks, Stamatis. That was
Okay.
terrific, and yeah, a lot of food for thought for everyone. Just a reminder that the talk has been recorded and will be in the YouTube channel of the international seminar series on chromosomal instability. So we'll circulate the link as well. And yeah, please join us in November for the next talk by Aelith. And looking forward to that. Have a nice weekend, everyone. And yeah, thanks a lot once again, Stamatis, for a great talk. Thank you very much.
Thank you all. Bye-bye.